live at what minute? We just start at 0 minutes, right? Chris? Okay. I think we came to this seminar. I would like to welcome everybody to Endogamy Now What? And we are thrilled to have Kelly Bergheimer with us, who fell in love with DNA many, many years ago, and is very excited to have that added with genealogy. So I'm going to let you go, Kelly. And
hey, great. Well, thank you so much. It's so great to be at Roots Tech. I hope you're all enjoying some presentations, getting some good excitement to your family history and your tree building. We're going to talk about endogamy for this session. And let me just remind you, there's no audio or video recording, no photos of the screens and that sort of thing. And so we're so excited you're here. And I know that you might want to take a picture of something, but please remember we have intellectual property. So let's talk for just a minute about what's endogamy. For those of you who aren't familiar, we're going to get into some definitions in just a minute. But endogamy is one of the tougher things about DNA analysis. Endogamy, along with kind of the pedigree collapse, the multiple relationships, those are the things that bring challenges to DNA analysis. So let's talk first about what endogamy is. Endogamy is marrying or partnering within the limits of a local community or an area. So this is Riedlingen, Germany, where my husband's Bergheimers are from. And generation after generation after generation, they didn't go anywhere. They were just having marriages and children in the same place over and over and over. And so that's kind of like this. Marriages and children, intermarriages and children. And they happen to be German and Jewish. And so they were culturally, religiously, and geographically having children over and over and having multiple marriages within this community. So that's what endogamy looks like. Then we have what's called exogamy, and it's not very often talked about, but it's marrying or partnering outside the limits of a local community. So let's say, for example, one person is Canadian and their partner or spouse is Peru. So when you think about Peruvian and Canadian, those are very, very different cultures and communities and DNA pools, so to speak. So the likelihood of there being interrelationships between those families is pretty low. Not zero, but pretty low. So pedigree collapse, many of us have. So if you have ancestry from, let's say, for example, the US South, or if you have another one of those small communities where sets of brothers married sets of sisters. I have this happening in my family, my Tennessee and Kentucky families. And so pedigree collapse is when two related individuals have offspring and there are fewer ancestral couples in their tree. So sometimes we talk about it being a narrow tree or a skinny tree. So instead of maybe having 8 particular ancestors, you only have 6, something like that. So this is what it looks like. I know a lot of people like to see what does this look like? Well, let's say that we have a set of 4 brothers that married a set of 4 sisters. Okay? The brothers are related to each other. The sisters are related to each other, but they're not, the brother and sister are not related to each other. So, but their children have lineage from these two lines, and you notice how the tree is going to get skinnier, right? So pedigree collapse over many generations can turn into endogamy, but some of us have a little bit of pedigree collapse in this particular line, maybe some endogamy over here. So DNA is just often that mixed bag of all kinds of different situations. And some of us have what are called multiple relationships. So two matches might share multiple ancestral lines with each other. So, for example, I have a set of half first cousins, and my half first cousin Bill, his mom and my dad are siblings. So, Adeline and John are siblings, and Bill and I share a common grandmother. But Bill's father is related to my mother. So, we have a secondary relationship where his dad and my mom have a common second great grandparent. So that's a multiple relationship. So we're going to see probably in our ancestry results, if we're looking at our DNA match list, the matches that Bill and I share are going to say both in many cases. And Bill and I say both as matches because we have two different relationships. We have a maternal and a paternal relationship. So that's what multiple relationships looks like. And all of these complicate our analysis. So let's talk about how to visualize endogamy. How do you know it? You have maybe duplicate ancestors in your tree, and I'll show you some examples of these. Ancestry side view is telling you both as relationships. Sometimes matches share DNA in many ways, and I use the leads method to kind of sort those out. And then clustering tools are showing lots of gray. So let's look at some visuals of what you might see if you have endogamy. So these are some of my friends. I kind of sorted through some submissions from friends of mine. And this is one of my friends who has a duplicate set of Landrums in her family. And so she's trying to sort through all of them, but she's seeing these on her paternal side and her maternal side. And that's very common with pedigree collapse. And if it happens over and over and over generationally, it can be endogamy as well. This same friend of mine also has Lee lines that are converging. She has Lees on paternal and Lees on maternal. So your family tree might be giving you clues. Now, again, not all Lees are related to Lees, right? So we still have to do some research to see if Reuben and Nancy are closely related to each other. This is one of my daughter-in-law, and she's Mexican. And so if you see here on the far right, I've kind of outlined in different colors the surnames that are repeating over and over and over between her paternal line and her maternal line. So she has Casadas and Perez on both sides. And so this is the kind of thing we see in a community where there's a lot of intermarriage. We'll see those names repeat and even those same couples repeat. So we've got this set of ancestors here on a paternal line and that same set of ancestors on a maternal line. So these are small villages in Mexico where people intermarry, or at least generationally they did. But she came all the way to America to marry my son. And so they have exogamy, at least in terms of their own children. So the children have half endogamy. They have mom's endogamy, but not dad's. This is a friend of mine who is South African, and she was showing me in her lineage what all the surnames are. And I've kind of anonymized them a little bit, but how she's got all these duplicate surnames. And she's part of founder families that come from South Africa. And so she's seeing those same surnames over and over and over in her family. In ancestry, we can see oftentimes where we get a hint that we have DNA on both sides. It might be easy for us to dismiss that one of those is wrong. Of course, it's just my maternal line or just my paternal line. But dig a little deeper. If they're telling you there's matches on both sides, you really need to look into that because it is probably correct. You can also tell if you have endogamy when you have large numbers of matches. And I know this is a little bit small, but all the way at the bottom under unassigned, this is my daughter-in-law's test, I think. No, this is not. This is my son-in-law's. And he has 123,000 unassigned matches. Very, very common with different groups that have endogamy, like the Ashkenazi Jews. So if you look at how many unassigned and how many both sides there are, really good clues that you have endogamy. And if you see in the ethnicity that wide swath of Jewish ancestry or admixture, biogeographical regions, whatever term you'd like to use for that, you're going to see that the Jewish group spreads across quite a big area. Now, if you do leads charts, and if you're not familiar with that, you might not really understand this, but let me just give you a couple of sentences about it. You can use a chart called the leads method to separate out your matches into family groups. And when you normally do the leads chart and you do not have endogamy or severe pedigree collapse, you see distinct family lines. When you do have endogamy, what you will see are these groups of people who are all interrelated to each other. So if you look here at the 229 centimorgan match, you see they're in the yellow group, the blue, the green, the red, and the purple. That's an endogamy key right there, that you've got someone who's showing up in all the DNA groups. They're sharing DNA with all these different ancestral lines. And again, this is my daughter-in-law's Mexican and indigenous lines. This is another example of where you see all that crossover. This is, again, a friend of mine from South Africa, and she just has people sharing and sharing and inter-sharing. All different kinds of lines are intersecting. If you're using clustering tools like MyHeritage has an auto cluster tool that's amazing. And when you have on the left, when you do not have endogamy or you have very little multiple relationship or pedigree collapse, it looks a little bit like what's on the left. This is my mom's on the left. And these clusters or groups are all interrelated to each other and likely lead back to a common ancestor. What you see on the right is you see this huge group that's not very tight, if you know what I mean. Lots of white space and lots of gray around it. And chances are you're dealing with an endogamous group if this is what your chart looks like at MyHeritage in the auto clustering tools. Here's another example. This one on the left is, I believe that one is one of my in-laws, but on the right, you can see there's all that gray, all of those interrelationships. So there are some things to keep in mind with endogamy. So what we're here to talk about is some strategies, some things you can do. So now that we've talked about how you can maybe, I see it, I know I probably have it, what can I do next? So for endogamy, genetic cousins tend to share more DNA. Sort of like when your side view mirror says objects are closer than they appear, your matches are going to appear higher in your match list than they really are. And so one thing that we look at to see if we can parse out where do we really connect with them? If they're reading as a second cousin, according to the DNA tests, maybe they're a third or a fourth. And how do we know that? So you can divide the amount of centimorgans by the number of segments to look at the average segment size. Now that's something you'll have to do at Ancestry or the companies that don't give you segment information, but you can also look at the chromosome browsers and the companies that offer that. So I'll show you some examples. Larger segments with higher centimorgan values will typically suggest that you share a more recent common ancestor, a little bit closer relationship. So let's do some examples of this, of what kind of metrics you want to look at. So the Shared Centimorgan project at DNA Painter is the go-to, right, for figuring out how all these folks are related to us. And there are ranges of DNA sharing, but those ranges capture endogamy because we're getting almost double doses of things, right? So we're getting extra DNA from these relationships that we wouldn't necessarily have otherwise. And so they're appearing closer to us than they are. So I have an exercise here. I just wanted to show you a little bit. This is my daughter-in-law's matches, and I chose all 5 of her 53 centimorgan matches because I wanted to illustrate sometimes what we have to do with DNA analysis is figure out what is it that we can do with the time that we have, right? We need to be efficient with our time. And so in DNA analysis, sometimes we need to kind of pause a little bit and say, you know what? Which are the matches we're going to focus on? So we've got Catherine, who shares 53 centimorgans across 4 segments, Maria, the same amount across 5, Mercedes across 3, and LF, managed by Maria, shares across 5, and Rebecca shares across 4. So what does that mean? If we take 53 and we divide by 4, what does that mean? So that gives us some information that we can go on. We also know longest segment. Most of our companies will tell us the longest segment size. So the longer the segment size, the more important that can be. But again, we're still pretty low numbers here. So we're not going to get too excited. But let's just make a chart. So Catherine, if we take the total amount of centimorgans and we find the average segment length, and then we look at the difference between the weighted and unweighted. So Ancestry gives us a weighted and unweighted DNA, and we look to see how closely those align. And then we look for the longest segment size. There are several ways we can decide where to start. Now, that doesn't mean we aren't going to look at all of them. We probably will. And what if one of them has a really great tree? Well, we're going to focus on that too, right? So this is just kind of a guide of what to do. So if I look at the longest segment length, I would be looking at Mercedes. She's the one that I want to look at. She might have a possibly closer relationship, maybe a half one generation closer. Hard to say, right? Hard to say. This is all ranges and guesses. And she's also got an average segment length that's higher than the others. So Mercedes might be a good place to start. But you know what happens, right? We go to look at Mercedes and she doesn't have a tree and we don't know where she fits and no names look familiar, all of that kind of thing. So this is just a guide that you can use. We also have Rebecca, who's got a 27 centimorgan segment. But we also have this Maria and LF, managed by Maria. So those are 2 people who are somehow related to one another. So they may be parent and child, they may be sisters. One of them's interested in DNA, the other one's not. So you want to look for those managed bys that happen to have some overlap. So you could have 3 different directions where you start. So we're going to talk about some endogamy strategies that you can use here along the way. And remember, we only have an hour, so we can't get too deep into everything, but I want to give you some things to look for. And your handout has lots of resources where you can follow up. So let's talk first about weighted versus unweighted segments. So this takes us into a topic that's called IBS and IBD. So for those of you new to genetic genealogy, you're like, what? So IBS is identical by state and IBD is identical by descent. Those are 2 different things and they're very, very important to understanding our DNA results. So let's talk about them for a minute. Ancestry uses a tool called Timber and gives you a weighted and unweighted value. So unweighted DNA is pre-Timber. It's the total amount of DNA you share. But what's important, especially about endogamy and heavy pedigree collapse, is that you can have that extra sharing, and you can also have very, very small segments that just lead you back to a country or a geographical region or Judaism. So unweighted and weighted are 2 different ones that you want to pay attention to. At Timber uses 90 centimorgans to kind of kick in, and Timber removes all those small segments that are likely identical by state and not descent. So what we want are descent segments. We want things that came from particular people, not just things that segments that came from particular regions or groups, because we want to find our ancestors, right? So we want to be really careful with small segments. There's a very, very important topic to cover in greater detail. But for non-endogamy, we want autosomal to be less than 7. We're just not even going to look at. 7 centimorgans is that tipping point where half of your matches are true matches and half of them are false. And for xDNA, it's even higher. And I'll show you why in a minute. But xDNA, we want to set the threshold at 15 or maybe even 20 centimorgans. We just want to be really careful that we're not down in the weeds in the small segments. So let's talk about why. Let me show you how the testing company is reading your raw DNA. So I don't know if you ever downloaded your raw DNA results and looked at them, but there's a zip file that you get, and it's just a sequence of base pairs. And so let's say that I have my mom's DNA and my dad's DNA, and I have a cousin match, and I want to know what's that segment and who is that segment matching. So this cousin has a segment that also matches my father. That is identical by descent. So this cousin, my dad, inherited it from an ancestor that they have in common. So that's identical by descent. So if the segment meets the threshold, so it's of a certain size and this cousin is in my list because we're shared matches, this is identical by descent and it ties back to a common ancestor. But when the testing companies are reading our raw DNA files, we have other things that happen. This is another cousin match to my mom and my dad. So this is cousin number 2. Well, cousin number 2 meets that threshold, but this is identical by state. So let me show you why. Because the testing company is reading and weaving back and forth between the copy I got from mom and the copy I got from dad. And it's saying, oh, look, here's a sequence, but it's not a sequence from one parent or a sequence from the other parent or a sequence from both. So identical by state is just one of those things that happens when you're looking at really small segments and you're matching things in the weeds. So just stay clear of the weeds. Remember, you're a subset of your parents. You cannot have a DNA match that your biological parents do not have. One of them has. So you're a subset of your parents. So for me, my identical by descent parents or matches that match my dad or match my mom are true matches. And then there are some out there that don't match my mom or my dad. It's a testing company weaving back and forth. So that's one way to know that you have a true match if you've got parents tested, or if you're the parent and your children are tested. So this is called phasing. Again, much, much more that you can learn about phasing. So why should you use phasing? Well, a real match, a true match, is identical by descent. And so that's family. That's what we're looking for. So the purpose of using that phase data is to kind of improve our odds of figuring out who these folks are. We don't need to find out the identical by state because they either go back to our same Irish or Scottish or South African, or they go back to a biogeographical region, or they're Jewish, just generally Jewish. So it could be something that broad, and we're not ever going to figure out how we're related to them. So I want to talk for just a minute about triangulation. Triangulation is a really misunderstood topic, but you have 2 copies of each of your autosomes, your 1 through 22 chromosomes, and women have 2 X's. So it's really important to think about who is matching who. So if one matches 2 and one matches 3 and one matches 4, is that triangulation? You know, if they all match each other, they share a most recent common ancestor, and you might be able to figure that out if they're a close enough cousin, like maybe a third, second, or first cousin. If they don't all match each other, some may be maternal and some may be paternal. So that's just something to look out for. There are many, many blogs and other great information online about triangulation. So you definitely want to learn more about that. So you want to test at every company. So I don't know how many of you have heard this before. It's not just us trying to get you to buy DNA tests. Every company has different tools and different things that you can use that might help you with your endogamous groups or your heavy pedigree collapse. So if you use as many matches as you have from all the testing companies and use the ones definitely that have segment information, that's going to get you the most progress moving forward. And of course, DNA Painter. You could write a whole, probably a whole month-long study on DNA Painter and how to use that for endogamy as well. So you can crowdsource your cousins. So this is kind of a technique that we use in a lot of different kinds of DNA analysis, building genetic networks, finding people who are all connected to one another. But you can crowdsource those cousins and see how they can help you. Multiple testers and segment data in particular for multiple testers are going to really help you to evaluate information. So if you've got first cousins, second cousins you're in contact with or you've found, and they're willing to do DNA testing, that's going to be really helpful to you to isolate what do you have in common. And of course, your own siblings as well. And unique segments shared and found through chromosome mapping can lead back to a most recent common ancestor as well. So if you haven't tested your siblings and you have endogamy, you need to. Try to get them to do it. Of course, we want them to be informed about the things that they'll find and that sort of thing. We don't want to do anything sneaky, but inferred matching is one of those powerful tools if you have sibling data. So inferred matching for a set of siblings is taking the power of their matches, the power of their DNA information, and combining it with yours. So my company matches and my brothers and my sisters matches, they're all of our matches, right? Because we all three descend from the same two parents. So I got a little different DNA than my brother did, than my sister did. They have slightly different ethnicity estimates, slightly different match lists than I do, certainly at the closer level. But as we go further down, they're going to have more segments that in common with other people than I do. And so we can use the power of siblings. Use them all together. It's a powerful way to make progress in your research. And my inferred matches are just amazing. I found so many relatives. Also, it's important you understand the variability and ranges of matches. So I call this like the high-low. So sometimes we have high and low sharing matches. Sometimes we have a lot of difference in who's in our match list. So I just made a spreadsheet here. This is not anything auto-generated, but I chose a match, KA, and I looked at who matches me and also KA, and who also matches Jody and KA and Todd and KA. And look at how many more matches my sister has. She has 8 additional matches that I don't have. My brother has 9 more that neither of us girls have. And because full siblings share the same parents and grandparents, these matches to my siblings are as all assumed matches to our common ancestors. So that's inferred matching. So definitely go and scope that out. And you can do this for any set of siblings. You can do it for your children. You can do it for your own siblings, your mom's siblings. You can choose any set of siblings and do research using all of their data. You can also do the leads method with a set of siblings, whether you're endogamous or not endogamous. So I make a leads chart for one of my boys and my second boy and then my daughter, and then I combine them into one chart. So that's a lot more work than what we have time for today, but I just wanted to let you know that you can do inferred matching in combination with other things, and the leads method is one of those. So what I can find is matches that my daughter has that my 2 boys don't. I can find matches that one kid has that the other 2 don't. And so when I look at them in aggregate, what I see here is this match Paul matches my daughter at 209 centimorgans. That's a nice, juicy match. But she matches my boys. He matches my boys in the 40s. So they're low sharing with my sons, but high sharing with my daughter. So you'll start to see those patterns of low and high sharing and no sharing that happens sometimes. And you might be wondering why my boys look so similar. They're identical twins, and that's why their numbers are very similar all along that chart. They're not exact. That's just the fun of identical twins, but they are very similar. So you can learn a lot about inferring the matches, and I've just shown you kind of here a bigger screenshot where we've got several different matches that my daughter matches nothing with people, but my boys do, and some where my daughter matches and my boys are low sharing matches. So, Y-DNA and mitochondrial DNA analysis could be a really good strategy for you with endogamy. So, let's talk about that. So, if you target test what's called your matrilineal line, your mitochondrial DNA line, so mom, and your patrilineal line, that's dad, that Y-DNA and mitochondrial DNA, you can isolate some family lines sometimes using those because my Y-DNA and mitochondrial DNA do not have recombination. There's no mixing of parental lines using mitochondria and Y. And so you can find a mitochondrial signature of each grandparent or a Y-DNA signature of each grandparent or great grandparent to differentiate possibly ancestral lines. Now, I will tell you this is expensive and time consuming, and sometimes it's a really long game. You have to wait till you get matches. But Family Tree DNA has just announced in the last week that they are releasing new mitochondrial DNA tools that will help us in our analysis. Very similar to the Y-DNA tools, the haplostory, and those kinds of tools that they already have available are going to be available for the mitochondrial side as well. Mutations in those Y-DNA and mitochondrial lines can help us delineate branches. And Johnny Pearl at DNA Painter has recently added mitochondrial and Y-DNA fields to DNA Painter. So as you're painting chromosomes and identifying ancestors, you can add in those mitochondrial and Y-DNA haplogroups to your paintings as well. So which DNA undergoes recombination? Which one gives us all the fits with endogamy? Well, autosomal DNA recombines and xDNA recombines. So those are going to be challenging, not impossible, but challenging. And then mitochondrial and Y might be a really good bet for you if you have a lot of endogamy to identify those particular ancestral lines. So I just pulled up my family chart and I just want to show you a couple of things because I think we get really narrowly focused on mitochondrial and Y. We think about I have a father patrilineal line and I have a mother matrilineal line. And that's it. No, we have lots of matrilineal lines and lots of patrilineal lines, depending on which ancestor we're looking at. So if I look at my great grandfathers, so I look at John Reese, I've got a patrilineal line there. I've also got a Calderwood patrilineal line, a Todd patrilineal line, and a Robbins patrilineal line. So I've got 4 Y-DNA testing lines that I could start with and get some differentiation there. I could also look at the matrilineal lines. So from me to my mom to my grandmother, my great grandmother, that matrilineal line along the bottom. What other matrilineal lines are there? Well, I have another. I have a father who has a matrilineal line. I have a grandfather who has a matrilineal line. I have another grandfather with a matrilineal line. So you're probably understanding at this point that there's lots of different matrilineal lines and patrilineal lines within your family. You only have one of each if you're male and just have a matrilineal line if you're female, but you can identify other ancestors, find descendants living in real time, and test them and see what kinds of haplosignatures you're finding. So how do I use Y-DNA results? Well, you could join a surname group or one of those projects at Family Tree DNA. You can also work on your genealogical tree, your paternal line, using other surnames of those matches that you find. So if you've Y-DNA tested and you see other surnames, definitely check them out. Look for locations. And if someone has the exact same Y-DNA as you, they're in your patrilineal line. And so you want to figure out how are they related. So for matrilineal line, very, very similar list, except there aren't surname projects for matrilineal lines. There are haplogroup projects. And you're going to want to work on that maternal line, that matrilineal mother's line. Look at locations. And if they're an exact match to you, that genetic distance of 0, they're in your matrilineal line. Now, your genetic distance of 1 and 2 and 3 are as well, but they're probably a little further back. There are some other caveats there, but we're not going to get into those too much. But there are some great tools that Family Tree DNA just rolled out in the last week. So if you've done mitochondrial DNA testing, go check out and see what they've posted for you recently. There is another thing that you can do if you have endogamy, and that's checking for runs of homozygosity. Now, homozygosity is a big word. So let's talk about what that is. It's where you have a strand of DNA, a piece of DNA, a segment that is on your maternal line and your paternal line. So your mom inherited it from a set of ancestors that is also a set of ancestors for your dad. So there's a piece of DNA that's being inherited on both sides. There are some tools that you can use to identify this, and one of those is GEDmatch. But let's talk for a minute about runs of homozygosity. There are regions of DNA where the maternal copy is identical to the paternal copy, that segment. Data is the same. Remember when we were looking at that raw DNA strand and looking at all those bases? So if a match matches you in that location, you want to double the amount of centimorgans to see how they're related to you for that segment, because we want to account for both copies of that chromosome. That's a little bit more advanced, but just something to keep in mind. David Pike has some resources for GEDmatch, the Are Your Parents Related tool. If you're not familiar with GEDmatch, it's a third-party tool where you can upload your autosomal DNA results, and you can use David Pike's tools to check for these runs of homozygosity to see how closely your parents may or may not be related. If your parents are related to one another, either closely or further back, maybe second great grandparents or thirds perhaps, you might see little segments of DNA that they have in common. So the Are Your Parents Related tool at DNAsci.com has a relationship predictor, and Kitty Cooper has some excellent blog posts on that for your Are Your Parents Related tool as well. So lots of more you can find out about that, and there should be some information in your handout as well. So let's talk about longest segment. This is something that will indicate whether or not someone's a little bit closer in relationship than you might think. So we can use some of our chromosome browsing tools. So DNA Painter has chromosome browsing tools, but our DNA testing companies do as well. So I pulled a match for my husband, Christopher. And so I looked at one of his matches, and I wanted to look to see how can I compare the segments they share in common. So at MyHeritage, you can do that with their chromosome browser. And so when I look at how their segments are related, they've got one long segment that's 25 centimorgans that they share, but then they've got these other three pieces, a 9, a 10, and a 10. So what do you think about that? Well, that 25 is a pretty good chunk, right? So I'm interested in that. I think this is a legitimate match. I could probably find the most recent common ancestor, but I don't want to go much below the 10s. And I also don't know if this is really going to be a fourth cousin, a fifth cousin, an eighth cousin. It's so hard to tell, because if there's endogamy, and my husband's family has some Ashkenazi Jewish, and if there is endogamy, then some of these segments might be very, very further back ancestors that have just been inherited because they happen to have Jewish ancestry. So my husband has another match, and this one's to someone named Rami. And so when I look at that in the chromosome browser, I see kind of a different story, right? I don't have a longer segment to focus on. 14 is the longest. And then I have these 10, 6, 6, 6, and 6. Well, that I'm going to set Rami aside. Sorry, Rami, but you're probably not going to be too helpful to me, because that 14 centimorgan longest segment could be a fifth, sixth, seventh, eighth, or more distant cousin. And so I'm probably not going to figure out how he and my husband are related unless I can find a whole group of people who all share those segments together. And that's a pretty long shot at this level. You can use that set of siblings to do what's called visual phasing. And so you can visually phase your siblings and develop a map of your grandparent lines. And you need a set of 3 siblings. They don't need to be your siblings. You can use any set of siblings. But again, we're harnessing the power of siblings. We're looking for recombination spots. And this is something that's very, very involved. Blaine Bettinger has done an excellent series on visual phasing, how to pull the data, how to interpret the data, how to find those crossover points or those recombination points where each segment ends that you inherit from each of your grandparents. And so it's pretty involved, but it's also very well worth it if you're able to accomplish it. So if you have sibling data, definitely check out visual phasing. Also, you can work on non-endogamous lines and then come back to endogamous lines. So that's what I did with my son-in-law, Mason. He has a whole line of his family, his paternal line, that is Russian and Jewish, and coming from what's now Ukraine, but what used to be Russia in Odessa. And so what I decided to do with him was to work through all of his non-endogamous lines, and then to come back and work on the endogamous lines. So I worked on his mother's family, and then I worked on his father's mother's family, and then I had what was left over as his father's family. So that's the way that I approached working on his endogamous family, is kind of clearing out all the ones who aren't, and then focusing on the ones who are. You can also look at in Ancestry's tools, they have the parent overview, and you can see whether or not those endogamous groups, such as the Jewish or the Russian Jewish, is coming from one parent or it's really coming from both. And so that'll give you an idea of how you can weed out mom's side and focus on dad. And even if you know that dad's mom is not Russian Jewish, you can sort out dad's mom and just work on dad's line. So some of those can give you some clues as well. So what do you need in your toolkit? Well, I've given you lots of things in your handout and some suggestions for today, but keep in mind, we have a really hard time in DNA with confirmation bias. We think we know what we know, and we're not going to be persuaded otherwise. We really have to think about things with the scientific method. You want to test in all the testing companies. Stay away from small segments, they're perilous. And doing that descendancy researching, working forward in time to find your DNA matches and who they descend from is going to be most helpful. Remember the scientific method. I know some of you had some science classes where you talked about this, but it may have been a long time ago, as it was for me. But the scientific method is really important. We have to kind of take off our genealogy hat a little bit, our bias, and we want to try to approach it more as a scientist and form some hypotheses and ruling things out and ruling things in. You know, science doesn't just accept this piece and that piece. We have to really look at what's called the body of evidence and come to a genealogical proof standard. There are probably some other sessions at Roots Tech about proof standards and scientific method and confirmation bias and all sorts of other things that you can do while you're here at Roots Tech this week. You need more than DNA for proof. You want to focus on the body of evidence, not just a few small pieces of evidence. Think of it like a detective building a case, right? The genealogical proof standard, there are 5 steps to it, and I'm not going to spend much time on this, but I want you to be aware that there is a level of proof required. It's not just that, hey, we share DNA and we have the same surname, so therefore we're related through this, it's more complicated than that, right? But citing your sources and writing things down and evaluating sources, all of those things, and resolving conflicts are all part of the proof standard. There are some promising tools and some information on the horizon, and I've listed a lot of these in your handout. There's a new-ish thing. I've seen it around for maybe a year or so called SMARTA. It might have been around longer than that, but boy, is it hard to keep up with everything, right? So shared matches, this is based on averaging different tiers of relationships. And so you'll want to look up Steve Taub, and he's written about his experience using the leads method with endogamy. And so the focus is finding the strongest matches, just like some of the other strategies we talked about today. And he uses an Ashkenazi Jewish admixture kit, and he uses Ancestry, and he looks for patterns in the clusters. And so he's done a lot of writing and work and has web information about SMARTA method. He has some tables in his blog, different relationships of tiers, probably solvable, possibly solvable. So he's doing that same thing where he's looking at the data and saying, this is where I should probably spend time. This is where I might be able to find an answer versus this answer is going to elude me for a long time. And so we only have so many hours in the day, right? And we can only spend like 20 of those doing genealogy. So we had to be focused on what we're looking at and what we're solving. There's also something promising in the Shared Centimorgan project with the longest segment methods. So if you want to go to Facebook, Blaine Bettinger's group has a big discussion about longest segments. He has a PDF there with data and histograms and all kinds of things about the meiosis groupings. So that may be a little bit deeper than a lot of you want to go. But here's what he wrote about it on Facebook. And it's not a relationship probability predictor, it's for analysis only. But he's looking at segment data, longest segment data. And so I expect that he's going to have some really good simulations, some large scale kind of information coming to us pretty soon. I think the first time I saw this was last summer, spring or summer. Another promising tool, Banyan. You've probably heard of Banyan DNA. They're here at the Roots Tech Conference. So if you're attending in person, they have a website. There's some great information, great teaching that they do over there. So Banyan DNA is working well for pedigree collapse in multiple relationships and being able to draw those relationships out. The future, we hope, is that the tool will be getting better and better in terms of endogamy. So definitely follow them and see what you can find out at Banyan. But it's great for graphing or diagramming those complicated pedigrees that we have. There are some other resources that I'll mention here. Your DNA Guide. I work with Diane Southard at Your DNA Guide. I'm her director of education and help her develop courses and curriculum. And we have an excellent blog. We also have an endogamy course. So if you're interested in that, we're going to be launching that again in August. So get on the wait list. Go to the website, get on the wait list. Legacy Tree Genealogist has a fantastic blog. And I know Paul Woodbury and some of the other genealogists on their staff, their DNA staff, write some amazing blogs there. So check them out. DNA Explained, Roberta Estes has a blog. She gets deep in the weeds, y'all. She's got all kinds of great information talking about segment analysis, all kinds of different tools. She is an expert and has a new book about family tree DNA and how to make the most out of that, Y DNA, mitochondrial DNA, that sort of thing. She's a great resource. Segmentology.org has some great resources. DNA Painter blog, and especially about endogamy, some of the blog posts by Laura Diamond. You definitely want to check those out. And Blaine Bettinger's Facebook group, Genetics, Genealogy Tips and Techniques, is where you'll find that information about longest segment and the PDF that he's working on and some of the things that are in the works there. And you can keep yourself current. You can do this. Yes, after telling you how hard it is for the whole time, you can do this. If you're good at recognizing patterns and you have logic and you're organized and you're persistent and you're patient, and maybe have a little bit of luck, you can do this. Endogamy is not easy. It's really not easy. Work on your non-endogamous lines if you have them first, and practice identifying all those folks in that first, second, third cousin categories, and then work on the endogamous lines. But I hope this has been helpful to you. So don't forget to give feedback in the app. And if you want to look for me, my email address is on the handout, but also I have 2 books available on Amazon, and I have a LinkedIn profile, and my profile there is for writers, etc. And you can find me in all of those spots. So I think we're going to do some questions.
Thank you, Kelly, so much. That was really amazing. I do have some questions.
Okay.
Would you explain what's the difference between weighted and unweighted segments?
Yes, so unweighted segments is how much DNA you share with someone. But weighted means that the company has pulled out those teeny tiny pieces that are probably not going to be helpful to your research and are maybe helping it look closer than it appears, the relationship. So, one of the examples that I showed you with my husband's DNA was that match that he had that had all those six, 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 seven. That's someone that all of those sixes and sevens, if they were at Ancestry, that test, they would probably be pulling those out and just focusing on that one chunk of DNA that's relevant. The others just lead back to biogeographical regions.
Okay. Next question is, does 23andMe have a working chromosome painter?
They did. They have not recently, and they're here in the vendor hall. And they also, I think, have a virtual vendor booth as well. So those are the kind of questions you might want to ask them. I have not seen it return, although I'm hoping, right? Because the chromosome browsers are so important to our analysis and especially for endogamy. But as of the last time I looked, sometime last week, I did not see it. I did not see it in my testing company site.
The next question is, how do you use DNA results from other companies if you want to compare them to Ancestry?
That's a great question. So that kind of multi-kit analysis can be a real challenge. I'll tell you what I do, and I do the leads method a lot. And so I make a leads chart for each of my testers. So I have one for my dad and my mom and then I have a one for each testing company. So I have my leads chart for Ancestry, my leads chart for my heritage, and so on. And then I combine them once I figure out which group corresponds to which group. So which group number at Ancestry corresponds to which group number at MyHeritage, then I can combine them. If you're not using the leads method, one thing you can do is you can make a list of your first, second, third cousin matches, close family too, and first, second, third cousin matches. And you can do this on a spreadsheet, piece of paper, and then go looking for them at the next company. So start with Ancestry. That for most of us, that's probably where we have the most matches. Not everyone, but a lot. And then go look at MyHeritage. Is there anyone in your Ancestry list who's also at MyHeritage? And if they're not, add them in. Add them in at the approximate cousin level. So with my second cousins at Ancestry, I'm going to add in my second cousins at MyHeritage and so on. So you can build that yourself. There's not really an automated tool that will do that too well. So hopefully that helps.
What is the difference between the leads method and the chromosome painting?
Oh, okay. So the leads method is a spreadsheet or a piece of paper. And what you're doing is you're looking at the shared matches that each of your matches have. And so you're looking at how you match someone else and who else matches them. And so you're dividing your whole set of matches into groups of matches and looking to see who is the common ancestor that they have in common. That's different than using the other company tools like a chromosome painter or a chromosome browser. So chromosome painting is what you would do at DNA Painter. Chromosome browsers are what's available. Used to be at 23andMe. They might bring it back, but we have chromosome browsers at Family Tree DNA and at MyHeritage. I showed you the MyHeritage ones today, but they're all basically the same. And so that will tell you what segments you have in common with someone, but that doesn't tell you how you're related to each other. That just tells you the segments. So you have to go to the chromosome painter and take all of your match data and apply it there to see if you can form those groups that lead you back to a common ancestor.
Okay, thank you. There's another question. Can you do inferred matching with a half sibling?
Yes, of course. So you can do inferred matching with a half sibling, but that's only going to tell you about one of your parent lines, right? So they're either a half sibling to you through your dad. And so that would give you your grandparents on your dad's side, your great grandparents on your dad's side. or maybe they're a half sibling to you through your mom. So that would give you your maternal ancestors. So, yes, you can definitely use the inferred matching. They're just not going to match all of your match list because you don't have two parents in common, just one. But you can definitely capitalize on their data.
Okay. Is IBS segmentation potentially why I or other folks with endogamy have so many shared and
Yes.
unidentified matches on Ancestry?
100%. Yes. So, identical by state, IBS, is the reason why you have so many matches and so many matches in the weeds. IBS can happen for a lot of different reasons. There can be what's called pileup regions that are specific to families, specific to groups. You can learn more about that at ISOG's website, the International Society of Genetic Genealogists. I know I'm throwing a lot at you today, but if you just search for pileup regions and genealogy, you'll be able to find it. So some things are related to that. Some are just small biogeographical pieces. So I've got a piece of DNA that comes from Ireland, and so does this person and these other 10,000 people who all came from Ireland. So it's way far back in time. We're not going to figure out who that descent line is. It just happens to be because we're Irish or because we're Jewish or because we're Cajun. So that's the importance of it. It just is able to give you a way to set aside those matches that either you're not going to be able to figure out or you don't have time to figure out. We only have so much time, right? So focus on those closer matches.
Another question. In the example of better matches to go with,
Uh-huh.
what if you have a DNA match that is 122 centimorgans, 5 segments, which makes the calculation 24.4? Then the difference between the unweighted and the weighted is 122 minus 122, which will equal 0. Is this still a good match?
So what I did was I used the 54 centimorgans for all of those matches, all 5 of those matches to my daughter-in-law. They were all 53. I didn't use the unweighted. I used the weighted. So they were all 53. So that may be where the confusion is. But if you watch the recording or go back and look at the slide, they're all 53 centimorgans that I used.
If imported test results to the different testing companies, do you still need to actually test at the different companies?
Well, that's a great question. So, what most of the testing companies we have, the top 5 anyway, that we talked about here today, and the other lectures that you're probably listening to or talking about, they all are testing relatively the same 600 to 700,000 markers in your DNA. So there's pretty much a one-to-one correspondence. Some of the companies use what's called imputation, and what that does is that kind of fills in the gaps and makes a very good estimated guess. And so if you transfer your DNA and there's not that one-to-one correspondence of the markers, imputation might come into play. And you can read more about imputation. I There are lots of factors that go into DNA testing. Finances is one of them. And so if you need to transfer rather than test at the company sites or the company DNA testing companies, that's completely fine. But I always recommend if you test at their site and you test with their and use their tools, you're going to have just a little bit better outcome than if you transfer in most cases. And there are some testing companies that don't allow transfers. Ancestry, 23andMe don't allow transfers. So,
Thank you for that. I have one more question.
okay.
Can you explain how I have more DNA to one person than either of my parents?
Yes. So you cannot share more DNA with a match than your parents do. So a lot of that is false matching. A lot of that is IBS. It's smaller segments. So whatever your parents are matching them. So let's say you have a match that's 100 centimorgans, but your mom is only matching that person at 50. Then 50 is what you go with in terms of predicting the relationship. Now, if you have endogamy, that combination of what you're seeing in your DNA results may be a combination of what mom shares with and what dad shares. So it could be a combination of that. So, but you cannot have DNA that you share with someone that your mom or your dad or both don't have. So some of that is noise, false segments.
Okay, we have some questions that are very specific, and I'm thinking that they can contact you directly, right, Kelly?
Well, yes, I don't do any client work and I might be able to do some short answers, but as far as taking a deep dive into someone's research, what I would recommend is that you find a course on endogamy and you learn how to find your answers. And so there are several courses on endogamy, and the one that I use is obviously at Your DNA Guide, where I work, but there are lots of other places where you can learn more. There are researchers for hire at the Association of Professional Genealogists. They have a website and they have people who specialize in endogamy in particular. And so I would definitely recommend that you hire someone. I can do short questions through email, but if you send me your family tree and your whole family history, I'm not going to be able to answer your question for you. It just takes too long for me. Sorry.
So, guys, we only have 30 seconds in this webinar. We are very happy to have heard this. Thank you so much for all this valuable information.
Yeah, you're welcome.
And we hope that you're having a wonderful time here at Roots Tech.
Yes,
See you later.
absolutely.
Yes, thank you.
Thank you.